Greetings everyone, let's jump right in. Polyploids are everywhere. They occur in almost every taxonomic group. Even more interestingly, polyploidy or whole genome duplication often precedes large diversification events. Perhaps the best example lies here in the angiosperm lineage, showing evidence of multiple episodic events of polyploidisation, often followed by massive diversification. This really brings us to ask the question into the evolutionary significance of polyploids. Polyploids are often different to their diploid parents in nature. They may also, after formation, show immediate isolation and differentiation to diploids. As can be seen on this extract, polyploid-induced isolation has a very strong effect across many traits. But its significance to evolution has been debated with mixed evidence across various taxa. Perhaps the most prominent phenotypic effect of whole genome duplication is the Gigas effect. The Gigas effect refers to the enlargement of traits as a direct result of the increase in the DNA amount inside of cells. After formation, polyploids are often faced with minority cytotype exclusion, as they are in the minority to diploids, but the Gigas effect brings about a host of changes and potential fitness advantages, enabling polyploids to escape this exclusion and successfully establish. The Gigas effect may cause heterosis in polyploid individuals, but more so, with larger cells and organs, polyploids may exhibit slower growth rates and experience changes to metabolic pathways. Slower growth rates and metabolic changes may increase a polyploid's nutrient use efficiency and increase environmental tolerance, enabling polyploids to establish novel niches to diploids. Slower growth rates may also cause a delay in flowering phenology. This could lower pollinator competition, may enhance attractiveness to pollinators, or polyploids may even make use of an entirely new pollinator and seed dispersal network than diploids do. Extracts from Porturas et al. In 2019 shows how prominent the Gigas effect is for polyploids. Across a host of traits, the Gigas effect has a very large effect size, and this effect size remains consistent. But whole genome duplication doesn't always lead to the Gigas effect. Although the number of cases are in the minority, some polyploids exhibit no difference in size to diploids, and in a few cases, polyploids were even recorded to have smaller traits. Why do we observe this variation? Is it something that's only isolated to a few groups? If polyploids are reliably larger, the Gigas effect could be a useful field indicator of ploidy level without timely and expensive methods like flow cytometry. Furthermore, if the Gigas effect is a reliable predictor of ploidy level, given all the ecological advantages associated with it, it could be a useful tool when working with variable lineages showing prominent cytotype diversity. And so, speaking of a variable lineage, Oxalis is a very large lineage with roughly 500 species globally. The genus is very well represented in the Cape with more than 230 species either described or awaiting description. The genus provides an interesting study system given its context. It occurs in the species-rich Cape floral kingdom. The Cape has a high species diversity, but very low levels of polyploidisation. The Oxalis genus, in contrast, has a very high occurrence of polyploids across the genus and within species complexes. As mentioned, it is also very variable, and especially in morphology and in size. Here's just an example of 4 species from this genus to show you the amount of variation in growth habitat, size, and floral displays. This variation is not restricted to between species comparisons though. Here is an example of Oxalis flava, a species with a high cytotype diversity, or meaning multiple levels of polyploids, many polyploids, and it shows incredible morphological variation. If polyploids show evidence of the Gigas effect, and this is linked to increased fitness, and given the high cytotype diversity and occurrence of polyploidy in the genus and in species, it could be possible that polyploidy is a driver of this variation. We therefore ask, is there a clear Gigas effect and signal in Oxalis looking across different species? And secondly, if we do extensive sampling within species, do we see an enlargement of traits with ploidy level? Which traits, if any, are correlated with the Gigas effect? Polyploids could also benefit from increased levels of self-pollination and clonal reproduction. And using Oxalis purpurea, a known local and globally invasive weedy species, we can also measure the ecological differences associated with polyploidy and the role it potentially plays in promoting invasibility of a polyploid. So for the first chapter, we asked this question: Do we find a clear Gigas effect signal at the genus level in Oxalis? We measure pollen diameter, stomata length, and cell area, as these have all been previously correlated with the Gigas effect. Using the Oxalis Living Research Collection at the Stellenbosch University Botanical Gardens, we sampled 65 accessions from different populations for 24 species. At least one diploid polyploid pair was sampled for each of these. All determinations for ploidy level were previously done at the Institute of Botany and the Czech Academy of Sciences. Here's a quick summary of the equipment and software used in this study. Oxalis has a tristylus reproductive system, meaning that each flower has a reproductive structure divided into 3 levels or whorls. Pollen sizes differ between anther-whorls of different levels, and because of this, we had to sample from a common anther-whorl for each of the diploid-polyploid comparisons within a species. Pollen was removed and mounted and stained for viability using the Alexander stain, and pollen grains are then photographed at 400 times magnification with 30 grains quantified per plant. For stomata and epidermal cells, we use the epidermis from sepals, as many Oxalis species form balloon cells on the leaflet epidermis, rendering stomata almost invisible and making area measurements inaccurate. Therefore, using one flower, we removed 3 sepals, and then 10 stomata and 20 cells were counted for each of these, amounting to 30 stomata and 60 cells per plant at 400 magnification. For all 3 of the response variables, we modeled for ploidy level in a generalized linear mixed model context, using accession and species with a random intercept and slope model. For pollen data, we had to include anther-whorl as a fixed factor to account for pollen differences between the anther-whorls. The effect size for the Gigas has been shown to be very large, and it remains consistent across traits. Unexpectedly, however, we find a significant effect for polyploids, but it is extremely small. Even more important, using these 2 examples, Oxalis adenoides and ciliaris, we found that the effect is also very inconsistent across traits. These are just 2 examples, but this was a commonly observed pattern for our entire data set. Polyploids are significantly larger, so we think there might be some kind of Gigas effect playing a role, but the effect size is so small, and it is inconsistent with massive variations between accessions and across traits. So if there is a Gigas effect at all, why do you observe this strange pattern? Possible explanations are that polyploids might be different to diploids in this context, but a clear Gigas signal might get diluted by natural variation after post-whole genome duplication adaptation. The large effect expected might be an overestimate, and in Oxalis, the effect of being polyploid may just be very small. It is, however, very likely that we have too small a sample size within species. We therefore need to do extensive sampling within a single species. This brings us to the second chapter, which aims to do just that. We do extensive sampling within a single species, Oxalis purpurea, and we ask the questions: is there a Gigas effect in Oxalis purpurea? If yes, for which traits? Are there any ecological differences between diploids and polyploids? Unfortunately, we are still waiting on the final set of data for this last question, so I will not be touching on it in this talk. Oxalis purpurea provides an ideal system to test this. It is widespread and common. Here are some of the variations you might expect to see in the field. And the species has multiple ploidy levels and is a known invasive weed locally and globally, enabling us to test the ecological effect of polyploidy too. We did extensive sampling across the species' native range with 12 plants per population, amounting to a total of 20 populations. Bulbs were removed and replanted in 50 centimeter deep soil in a common garden setup. They were then left to acclimate and go dormant. Trait measurements remained roughly the same as in Chapter 1. Additionally, we also measured 13 floral and leaf traits. Flow cytometric determinations were done in Prague at the Czech Academy of Sciences, Institute of Botany, and at the University of Pretoria, Department of Plant and Soil Sciences. Our response variables were modeled in a generalized linear mixed model context, though we struggled using GLMMs as a result of the model assumption violations. We tried multiple error distribution families and relevant link functions. Significance, however, remained the same and consistent across model constructions. Once again, we find large variation in significance and effect size across traits. Ploidy is still significant for many of the traits, suggesting that there is a very small Gigas effect. Here is stomatal length distribution for each of the populations, the trait that most strongly correlates with ploidy. I want to highlight here on the screen in the red box is George population, which was the only population with a single polyploid sympatrically occurring with diploids. It is also the population with the most clear signal. This population has a unique, possibly derived morphology where tristyly has broken down into semi-homostyly. It could be that the signal is more clear in this sympatric diploid polyploid population if polyploidisation occurred more recently here than in other populations. So, there is some suggestion of the Gigas effect being present. In most cases, however, it is very small and remains inconsistent across traits. It is possible that the Gigas effect might just get lost in huge natural variation. But considering the overarching patterns across both chapters, we do not find overwhelming evidence to suggest a strong Gigas effect in Oxalis. Polyploids have a tendency to be larger, but not by much and not consistently. Considering the George population, it is possible that the Gigas effect might be temporary. It may be very pronounced in newly formed polyploids, but gets lost due to possible post-whole genome duplication adaptation. We can therefore not use size as an accurate predictor of whether a plant is polyploid or diploid. But this brings us to the question, is Gigas playing a role in maintaining Oxalis polyploids in the Cape? This will be a good focal point for future studies. I want to thank the following people for assisting with lab work in the most pressing of times. I also like to thank our Czech colleagues for all their flow cytometric work and supervision, and also Sine Tsangase from the University of Pretoria for her flow cytometric work on Oxalis purpurea. Also my supervisors, Prof. Leon Dreyer and Dr. Kenneth Oberlander from Stellenbosch, and Dr. Donovan Kirkwood from the Stellenbosch Botanical Gardens for their unwavering support, clear guidance, and strong mentorship. Here's also references relevant to this talk, if anyone's interested in looking something up. Thank you very much for listening. Are there any questions?